And I'm really, really pleased to welcome this evening Dr. Jean Ristaino, who is the William Neal Reynolds Distinguished Professor in the Department of Entomology and Plant Pathology at North Carolina State University. But more importantly, is she works on that incredible pathogen which devastated potato crops in the Irish potato famine in the middle of the 19th century and completely changed the world in which we live, both in terms of biology, but also in terms of politics and human migration. So her lab studies the impact of migration, recombination, and hybridization on the evolution of Phytophthora species, those things that cause late blight in potato. And she has developed these amazing techniques for the use of 150 and even older herbarium specimens to look at outbreaks in the past, but also ways of perhaps thinking about monitoring those outbreaks in the future. So I'm really happy to welcome you here tonight, Jean, and I'm really looking forward to hearing what's been happening recently, and I'm sure everyone is really anxious to hear the story. So thanks very much, Jean, and it's over to you.
Thank you, Sandy. I'm really excited and happy to be here virtually to talk to the Linnean Society and the many Solanum enthusiasts around the world who want to hear about the potato plague. And I've titled the talk this evening, "The Potato Plague, a Painfully Interesting Subject." And that's a quote from Darwin. It's how he described the disease. And I'll get to that in a minute. Let me see. Next slide here. So I want to start out by just saying there are many emerging pests and pathogens that are increasing globally. If you look at this chart that was published a few years back by Dan Beaver and Sarah Gurr, showing the rise in emerging pests and pathogen outbreaks on all continents. And there's FAO has emerging plant diseases on their radar screen when they talk about the future of food and agriculture. Prevention of transboundary and emerging pests and pathogens in food systems are a major threat to the future of agriculture. I've been working on Phytophthora infestans, the pathogen that caused the Irish famine, for more than 30 years. And it is not just of historical interest. This disease is a re-emerging disease globally in many areas of the world where there are food insecure farmers. I took this photograph of a smallholder in Honduras a number of years ago who doesn't have access to fungicides. So really host resistance is the only way to control this disease. And in many areas of the world, fungicides aren't available to control late blight. This is just showing some of the symptoms on potato leaves and the tubers and on tomato fruit and leaves. Now, the infected potato tubers are a major source of spread of this pathogen. It moves globally in infected potatoes, and that's how it moved in the 19th century when it caused disease in the US and Ireland. And the other thing about this pathogen that makes it a really good pathogen for causing epidemics is that it spreads with sporangia. These are the small microscopic spores shown here on a potato leaf surface, and they can spread hundreds of kilometers. They're airborne and they can cause disease from one field to another. So we consider it a community disease. It's contagious and it spreads from one field to another if the field is not treated. And potatoes will die within a few days if they're not treated. This is a picture I took in Eastern North Carolina a number of years ago. But in 1845, the Irish didn't have fungicides available, and entire fields of potatoes in the country, in not only Ireland, but the EU and the UK were destroyed. So late blight is really, as Sandy mentioned, it's kind of was one of the early diseases that founded the science of plant pathology. It impacted the food security of Ireland. This is a family, an Irish family, uncovering potatoes that have rotted to the ground, and the stores of potatoes are destroyed by the pathogen, and it was their only food source. And so it caused a national migration of people all over from Ireland and the death of over 1 million people. We've been interested in several different questions about this disease, including where the first outbreaks occurred and what strains caused the disease. I borrowed these images from Alston Burke's 1963 Nature paper. We know that the disease first occurred in 1843 in the US, 2 years prior to the European continent and Irish outbreaks. So late blight arrived in potato tubers around the ports of New York and Philadelphia. And then 2 years later, we see outbreaks around Flanders and then into Europe and over to the British Isles. This is just within a matter of months from August to October. So why was this disease so devastating to the Irish? Well, although other crops were grown in Ireland, mostly for export to England, the Irish people depended on this crop for their major source of food. An average adult male ate 9 to 11 pounds of potatoes per day, and it was really a major source of food. So when the crop was lost, millions starved. I took this photograph in Quebec in Gros Isle. It was a quarantine facility in Canada. Many Irish immigrated on what were called coffin ships, overcrowded ships. People were malnourished, had typhoid and cholera, although we didn't understand that germs cause disease, but they died en route. And in these quarantine facilities, they were placed in close facilities, and human diseases spread amongst the Irish immigrants. So there's actually a mass grave of 5,000 Irish immigrants in Canada, just from around near this immigration facility. And today, about 40 million Americans can claim Irish descent, including my family lineage on my dad's side a few generations back. So I took this photo up in Cambridge, Massachusetts, and outside Harvard University. It's a monument to the Irish famine. It says, "Never again should a people starve in a world of plenty." And unfortunately, that is not the case. People do starve in many areas of the world today because of food insecurity, either as a result of conflict, like wars, like we're seeing now in Eastern Europe, or climate change, many reasons. So food insecurity is still a huge issue. And I think this Irish famine is probably one of the first examples of a huge problem with food insecurity with dependence on a single food crop. So who were the suspects in 1843? In 1843, we didn't understand that pathogens could cause disease. Louis Pasteur had not done his work. The germ theory had not been published. We did not know microbes could cause human disease or plant disease. So what's very fascinating and wonderful about herbarium collections is that there are archival records and very beautiful drawings of sporangia of this pathogen on specimen labels. I took this photograph from a specimen from David Moore it's a Botrytis infestans on Anthocercis alicifolium. It was a shrubby crop, a tree that was plant that was brought into the Glasnevin Botanic Gardens. But it was uncertain if the potato crop was attacked because of the peculiarity of the weather, or was there a microbe involved? People saw microbial growth on the leaves and stems, but there was a big debate whether it was a consequence or the cause of disease. And it really took the detectives, the folks like M.J. Barkley, and then 30 years later, Anton de Berry, to understand that it wasn't the wrath of God or bad air, but a microbe, which we know now as an oomycete, that caused this disease. Anton de Berry named the pathogen Phytophthora infestans, which means plant destroyer. M.J. Barkley called it Botrytis infestans, but it was renamed several times. It was called Peronospora infestans, and then de Berry coined the term Phytophthora. So the herbarium specimen labels are not only useful for understanding, and archival material are not only useful for understanding the etiology of plant diseases, but also there were letters with these specimens. I read a number of letters written by M.J. Barkley to William Gilson Farlow. Some of those letters are housed in the Farlow Herbarium at Harvard. And they would draw pictures in their letters. In this letter, M.J. Barkley is trying to understand the sexual state of Phytophthora infestans. They didn't see the oospores. De Berry had described oospores, but they were only seeing one. We didn't really know there were two mating types and enough about the oospores, but they saw other spores of Pythium species that were most likely in some of the samples. So there was a lot of letter writing back and forth about the sexual state. And then this image just shows how wonderfully preserved some of these specimens are, and you can actually still see the lesions on the leaves. So About 5 years ago, I was invited to the British Ecological Society meeting to give a talk on some of my work, and I stopped by Down House to visit Darwin's home. And I had not been there before, but I was really interested in seeing his house, and I'd been reading and collecting information on Darwin and the potato famine. In 1845, Charles Darwin's Down House had late blight on the potato fields, and Darwin, who was writing letters to Henslow, his former professor at Cambridge, said, "I have to thank you for several printed notices about the potatoes. What a painfully interesting subject. I've just returned from home and have looked over my potatoes, and I find the crop small and a good many rotted into the ground." So even Charles Darwin's farm was not spared the wrath of Phytophthora infestans. And interestingly, Darwin, when he went on his journey into South America on the HMS Beagle, talked a lot about potatoes. I've read his journals, and there are many times where he talks about the crew stocking up on games and potatoes, and that pigs and potatoes are as plentiful as in Ireland and South America, and how Captain Fitzroy thought next to wild people, wild potatoes were the highlight of the trip. So this Darwin was fascinated by potatoes, and he collected them during his voyages on the HMS Beagle. I visited Chiloé Island in 2009 with some of my colleagues in Chile, and the wild land races of potatoes are still grown there in Chiloé. But Darwin collected wild potatoes, and this is a specimen I found in Kew back in the early 1990s of Darwin's, and it was collected in 1834. It was a Solanum called Solanum maglia on the label. And there's a second specimen in Cambridge, the herbarium in Cambridge. So what I was interested in was Darwin's role in trying to understand and control late blight. He had the disease on his farm. There's a glass house that's still there, and amazingly, they grow some of the same varieties of potato that were grown when Darwin was alive. So what he did in 1846, he had brought tubers back, and those tubers were stored at Cambridge in Henslow's collections. They took the tubers out and they grew them out. His cousin, William Darwin Fox, grew the potatoes, and then he wrote a report for the Gardener's Chronicle saying they fared exactly the same as the other kinds. They've blotched in the leaf and a few tubers decayed. So the potatoes that Darwin brought back from South America, the particular lines that they tested, were susceptible to late blight. And amazingly, late blight still occurs on Darwin's potatoes. It's a picture of me in a few years back, and I was walking through the gardens, and sure enough, I spotted late blight. Don Feaster of Harvard University, the curator of the Farlow Herbarium, and I wrote a nice review article about Darwin's role in studies of late blight, if you're interested in following up on that. But what was really fascinating to me, I went to Cambridge, Darwin Archives in Cambridge a number of years ago, and there were over 140 letters between Charles Darwin and a businessman in Ireland named James Torbit. And Torbit was trying, he was a businessman, but he was also doing some potato breeding, and they believed that the potato, they didn't understand the pathogen, and they were thinking, well, the potato is worn out. We need to grow it from true seed to eradicate the disease. We need to plant it beyond a range of infection with infected varieties. So they understood that this pathogen was spreading and there was a contagion. But what they didn't understand is that the seed tubers were really the source of inoculum. So even if you grow potatoes from true seed, you still can get the disease if there's infected fields outside of the area where the trials are. But Torbit wrote the 60-page treatise, and I found in the letters from Darwin to Torbit that he gave him 100 pounds of his own money to fund the studies on resistance to late blight. So that's a bit about Darwin's role in the late blight, and I refer you to that bioscience article to read more. But I've been very interested in using mycological herbaria to study epidemics of the past. And this is a picture of the old IMI mycological herbarium on the garden at Kew Gardens. It's no longer there, but these specimens that I first uncovered many years ago were really the source of a lot of the research that I've done over the last 20 years on Phytophthora infestans, samples from Kew, from the Farlow herbarium, and from the systematic botany lab at USDA Beltsville. And what's fascinating is they're really time capsules of the past of both extinct and extant organisms. You can study genetic structure of old and new populations. You know, studies of taxonomy have been used for Solanum specimens in the past, but actually the pathogens in these specimens can be used for studying the phylogenetics of pathogens and function evolution of genes over times, origins of populations, and of historical epidemics. And so some of our work that we've done over the years has kind of evolved. We first started out doing PCR from samples. This is a specimen from Kiger in Germany in the 1860s. You can actually see the lesion on the leaf. We did mitochondrial haplotyping using small bits of the mitochondrial genes. We've done microsatellites, and we've done whole genome sequencing. And now we're doing kind of some hybridization capture experiments where we can amplify specific tag genes and trace them through time. So herbaria can be used to study a number of different things and different questions, not only tracking outbreaks over historical time, but when did biological invasions occur in different host plants? Has climate change impacted pathogen distribution? And you can really capture baseline data on pest and pathogen occurrence by just reading and data mining the specimen labels. So we went through the microportal and found about 1,200 mycological specimens that contain Phytophthora infestans. And this map shows kind of the time frame when the red dots 1845 to 1900, and then many fewer more recent samples, but mostly up into the 1950s to 2000. But we have globally specimens, many from the US and Europe, from different time periods, and those we've used. We haven't used all 1,200. There's still more work to be done, but we've used a good chunk of the samples to track P. infestans. These are the kinds of questions we've been interested in addressing. First of all, what lineage caused the famine? There was a theory in the plant pathology literature that a US1 lineage, which was globally widespread in mid-20th century and late 20th century in the US, was the famine lineage because it was found on all continents. So the theory was the US1 lineage was the famine lineage. So that was a question we wanted to address. Was that true or not? Secondly, where did it come from? Did the lineage that caused the outbreaks in the US and Europe come from South America or from Mexico? There was a theory that the pathogen center of origin, there is a theory that the center of origin of the pathogen is Mexico. So was it the source of the outbreak inoculum? But we know that in the 1840s, potatoes were being grown and exported from the Andean region into both the US and Europe. We were interested in the pathogen's genome. I was part of a big team that sequenced the genome. We knew the modern genome size. We were interested in comparing modern and ancestral genome size and how changes in effectors, which are proteins that are responsible for virulence of the pathogen, have they changed over time? Were ancestral populations clonal or sexual? Were the historical genotypes still circulating? Did the same lineage cause disease in the old and new world? Was there more than one lineage that was dispersed in the 19th century? And did it go extinct? Did this lineage go extinct? So I'm going to tell you bits and pieces about all our data on all these questions. It was proposed, as I mentioned, in the early 1990s that the US1 genotype caused the famine and that it originated from Mexico, went into the US, and then spread globally and into South America and to the other continents. But those studies were all done with modern outbreak samples. No one had actually ever looked at herbarium samples to investigate this question. So when I was on a trip over to Ireland back in 1998, I went to Kew Gardens, found the samples from the Irish famine that are shown here in this window in the IMI herbarium. And we started writing grants, first to National Geographic Society and then to USDA NEFA. We were able to get some funding to do mitochondrial haplotyping. And we showed that it wasn't the US1 genotype, but a different mitochondrial haplotype altogether that caused the famine outbreaks. And in 2013, our team and another team actually did whole genome sequencing, Illumina sequencing from samples. And, you know, the question of whether that lineage went extinct or not was up for question. And I'll show you some data on that. We know there's lots of historical evidence and from ship records, and just from, if you read the Gardener's Chronicle in 1843, you'll see advertisements for buying your Peruvian potatoes or buy that guano. They were shipping potatoes and fertilizer from South America into Europe and into US ports. So potatoes were on the move at that time, and the steamship had been developed. So potatoes could move from South America to Europe in a very short, a much shorter period of time than they could have in any other earlier century when ships moved by sail. So I had a series of postdocs that worked on this early question, Carol Trout, Julia Hu, and Kim May. And we went into some of these early samples from 1846. We did mitochondrial haplotyping. And this is just a figure from our Nature paper and a mycological research paper. We were going after 3 different SNPs in the mitochondrial genome. And those SNPs allowed us to sort out whether it was US1, which is a 1B haplotype, or another lineage. And we found that it was the 1A haplotype, which is a distinct different mitochondrial haplotype than what was proposed. So we pretty much showed that US1 didn't cause the Irish outbreaks or the US outbreaks. It was a different lineage altogether. And it emerged later. We clearly found by going through all these samples all the way up into the 1930s and 50s that the US1 mitochondrial haplotype really didn't emerge till mid-20th century. So there was a shift from about 1843 to the early 1920s and 30s, we start seeing US1 come in. But previous to that, it's 1A mitochondrial haplotype. And then we fast forward a few years. This is a team that sequenced the pathogen genome from the Broad Institute, led by Chad Nussbaum and Sophie and Kamoon and Francine Govers and myself, a whole lot, Howard Judelson, a whole group of late blight researchers, Bill Fry. We had a big group of folks that worked on this project. And what we found was that this genome was highly expanded and it was a much larger genome than Phytophthora sogium remorum, and that the effector diversity, or these avirulence proteins needed to make the pathogen infect, there were many more of them in the genome of Phytophthora infestans, and they were in this gene sparse region of the genome. So fast forward another couple years, we decided to use that T30-4 Broad genome sequence as a baseline sequence, go back into the herbarium specimens and ask the question about genome size and avirulence genes in the pathogen. So I collaborated in this project with Mike Martin, who's now Trondheim University in Norway, and Tom Gilbert, who's at the University of Copenhagen. And we sequenced a number of samples from the from the European outbreaks. This T30 genome is the Broad genome sequence that was done in 2009, and US 8, 22, 23, and 13A2 are modern circulating genotypes of P. infestans. And what we found was that it was a highly supported monophyletic clade for the historic samples. The T30 genome was kind of intermediate between these other 2 clades, and that the samples from modern and historic time differed by more than 120,000 SNPs. And even between 1845 and 1889, we had about 12,000 SNP differences amongst the historic samples. So that suggested to us that this pathogen it was likely brought into Europe more than once, but it was also evolving for expanded virulence, most likely as our genes were deployed with the solanum, the potato breeding. So just kind of a little recap on ABR genes. This is a figure from Mike Martin, my collaborator, but essentially when there is a host resistance gene that's detected that the ABR gene interacts with, you have no disease. But if that resistance gene isn't in the host, you're going to get disease, and simplified. description. And there have been more than 20 AVR genes that have been cloned and sequenced, some in Wageningen, some at James Hutton Institute and John Ennis Institute. So people have been studying AVR gene interactions for a number of years. So we have that information and those gene sequences that we could mine out of the historic samples. And what we did was actually compare these inner rings are historic genomes, and the outer rings are the modern T30-4 and US 8, 22, and 23. And we found that the avirulent genes that are really considered important now for disease in modern populations were absent in a lot of the historic samples. So it looks like the genome had expanded over time, and there was an expansion of effectors over time. And so things that we recognize now as really important for virulence in the pathogen, they were not in some of the historic outbreak samples. So as we were doing this work, Mike and I and Tom Gilbert on some of our historic samples, Sophia Kamun's group was also sequencing some samples from Kew. And they sequenced the mitochondrial genomes and nuclear genomes out of a small set of samples. And they named the Famine mitochondrial haplotype as the ERB1 mitochondrial haplotype. And they drew this figure that suggested it emerged out of Mexico, caused disease in the US, then went into Europe, and then went into South America and the rest of the continents. This figure looks very much like a figure that Bill Fry would have published 30 years ago using RFLPs. So essentially, another paper also came out by Paul Birch and David Cook suggesting that this famine lineage went extinct. And in eLife, there's a figure showing this is the famine lineage. This is the US1 lineage. It spread globally, but the famine lineage went extinct. So immediately after we read that, we were like, I wonder if the famine lineage went extinct. We need to look at more specimens. So we started looking at more specimens, and we also in our work, we're studying Phytophthora in South America. And I want to talk about the evolution of species in South America. Phytophthora infestans is in a clade called the clade 1C. This Phytophthora ipomea, mirabilis, and infestans, these have all been reported in Mexico. But Phytophthora andina is only been reported in the Andes, in Colombia, Ecuador, and Peru. And it's on a number of selenum, different hosts, potassium, Brugmansia, and Ricardo Oliva and I and some colleagues from SIP wrote the species description to Phytophthora andina. So we decided to put Phytophthora andina into our phylogeny and compare that species genome to the historic genome sequences. And this is a figure for the mitochondrial genealogy and the nuclear genealogy from the Illumina sequencing that we did and published it with Mike Martin et al. In 2015. And we found that ERB1 mitochondrial lineage did not go extinct, that it actually persists in Phytophthora andina. That lineage, we picked up that mitochondrial lineage in this Phytophthora species that's only found in South America. We also found that we did the divergence time of that mitochondrial lineage, and there are 4 known haplotypes, 1A, 1B, 2A, and 2B, and they all, the ancestral mitochondrial lineage diverged in South America. These 1A and type 2A and 2B are South American isolates. And then the ERB1 lineage kind of diverged about 200 years back in time prior to the Irish famine. So it didn't emerge in the US, it emerged prior to the famine in South America. And when we put Phytophthora andina into the nuclear genome phylogeny here, this is maximum likelihood trees, we found that P. andina showed mixed ancestry with famine lineages. These are historic outbreak samples, and this is P. andina. It's only found in the Andean region, and it shared, it was basal in the phylogeny, and it had mixed ancestry. And the famine lineage was a sister lineage of P. andina. And then the US1 that I mentioned, that was the putative old lineage that caused the outbreak, we show it's derived, it's next it's diverged later than the famine lineage, so it didn't cause the famine, and the whole genome sequencing confirmed this. We also found that, interestingly, isolates from Mexico, all the basal branches in this phylogeny are from isolates from wild solanums. So that suggested to us that this pathogen did a host jump from wild solanums into Solanum tuberosum when it arrived in Mexico, and it could have arrived there on movement of wild solanums, which happened through the Andean region by native indigenous peoples. And then we found modern Mexican lineages, and these are modern US and European lineages. There's admixture between Mexico and the US and Europe, because the pathogen has moved around a lot in the recent 50, 60 years, and we get admixture of lineages here. And then the South American lineage that we tested in this particular study were most recent because they were modern imports from the EU. The Netherlands ships a lot of potatoes to South America, and the isolates that we had in this particular phylogeny were not historic isolates. These were modern late blight from South America, and they are all more recent introductions. So I mentioned in our previous work, we're showing kind of the evolution of and the ancestral relationships of the famine lineage to modern lineages. But we also, I mentioned earlier, the outbreaks were in the US before they occurred in Europe. So we decided to use microsatellites, which we use for modern P. infestans genotyping. They're 12-plex loci that we amplify and then type. And Amanda Seville worked with me on this, my lab manager, and we did the microsat genotyping, and we went through a lot of specimens, several hundred. And these small dots are 19th century European specimens. These are 19th century US specimens. And we found that the EU and the European microsats were identical. Same lineage. We did structure analysis. This is the EU-US famine lineages. Here comes the 1B later, and then modern South American, and then admixture of central US and modern Mexican isolates. So we now know we can use microsats, which are a little bit easier, less expensive to run on herbarium samples, or we can do Illumina sequencing, which is a little more costly, and the data sets are more complex to analyze. So the other thing Amanda and I have done, which we published last year, was we tracked the spread of this FAM1 lineage, and we found it did not go extinct. We found that FAM1 lineage present globally on 6 continents, even up to in Malaysia, and we found it like in samples from like the 1930s. I need to go back and look at the specimen labels, but they were, it didn't go extinct. It survived for 140 years. And what's fascinating is, once it arrived in the US, went over to Europe, the British moved potato seed. And with British colonization, you see that famine lineage show up in East Africa and in India and in Australia and in China. So British colonization potentially moved late blight. I just wanted to show this one of our older specimens from Colombia. This is the FAM1 lineage from 1913. So it's in Colombia still from this specimen label. This is from a Bogota sample. So we've done some of the largest population genomic studies to date on Phytophthora infestans. We showed that this HERB1 mitochondrial lineage diversified, probably multiple introductions into Europe. We've got P. andina as one of the ancestral, one of P. infestans. It hybridized with P. infestans and an unknown parent, likely in the Andean region. The most basal P. infestans like Haplotype survive in Phytophthora andina in Peru and Ecuador. Clonal lineages of P. andina and infestans diverged earlier than the Mexican lineages. The famine lineage was clonal. We're seeing one lineage globally, and it survived more than 140 years with British colonization. Our most modern aggressive lineages, like US 23, are mostly derived, and some others are derived either from Mexico or South America. And some other interesting things that I think I mentioned earlier, that we were able to show one of the early host jumps of P. infestans. This is that same specimen label I showed you earlier. This is Anthocercis ilicifolia, and Phytophthora infestans cause disease on that particular solanaceous host in the Glasnevin gardens. And the plant was actually introduced from Australia, but it became diseased in the gardens there when late blight was present on potatoes. And these solanum collections can be used to really study and track other diseases. This is a photo I took when I was visiting Sandy Knapp at the Natural History Museum in London, and many of the specimens have lesions, but we don't know if they're P. infestans lesions or others. All the work that we did previously was with mycological herbaria, where specimens were identified as having Phytophthora infestans in the solanum tuberosum tissue. But there is many more solanum collections globally that could be used to track what are these other pathogens, how climate change might impact disease, were there more than one leaf spotting disease causing trouble. Some of these could be early blight, some could be late blight. We don't know. We just haven't even begun to look at the larger collections. And then these data can be linked back not only to the DNA data, but now a lot of the specimen labels are being digitized, so you can look at the specimens to select ones that you might want to choose to work with, and then also mine the data on the labels. We've been doing some data mining work with archival records using PDFs. My colleague Laura Tatalsian and a group of us published just a meeting proceedings, but we're working on a paper on this where we actually went back into the patent office documents, were the early USDA documents during the US introduction of P. infestans in 1843 to 1850. And we're able to pull out through data mining, where they thought the pathogen came from, how they were controlling it, what varieties were being planted. And so using text mining and data analytics now, there's really powerful tools that could be used on specimen labels as well to track outbreaks. We just did a Google Ngram search, and we were able to actually capture, this is when we use the word potato disease and potato rot, we capture the 1840 outbreaks from Google Ngram searches. And later on, the disease was called late blight. And you can actually, this is just the number of hits from an Ngram search, but you can actually get a time series record of pathogen disease outbreak occurrences from some of the data mining tools. And there's more to be done on this. I visited SIP with one of Sandy's students, Gina Sarkinen, and some other folks at SIP a few years back, and we went up into the high Andean region. And I think the Andean region was not only the source of late blight, but it's also a source of host resistance genes. We know that there's germplasm there and wild solanaceous species that could be useful for breeding for disease resistance. And as I mentioned, there's novel species of Phytophthora that have been described. There's a couple new species, Phytophthora bataceae and uriae, that have recently been described. So there's more to be learned in the Andean region. And then also the indigenous knowledge from indigenous people on disease control. I visited a market when I was there and spoke with this woman who was selling potatoes, and we talked about how she controls late blight, and she was telling me about the soap they make to control the disease, and they make it from a local tree. So just some of that indigenous knowledge is that these growers have known for centuries how to control this disease. And I think there's much more to be learned from working in the Andes. I do want to end by talking about modern late blight epidemiology, because we do spend a considerable amount of our time working on modern outbreaks. And in 2009, we had a big outbreak in the US caused by a strain that we named US 22, and it spread from tomato transplants, from tomato fields into potato fields. And it was all over the newspapers, New York Times, about a tomato disaster. A group of us, led by Howard Jettleson, were able to get a project funded called USA Blight, and we still maintain the disease surveillance portal. We map outbreaks as they occur, and we're developing new diagnostic tools, including LAMP assays. We're genotyping strains using microsatellites, and then Bill Fry's group had developed some decision support tools for control. So we are continuing to provide information on how to manage this disease. Some lineages, modern lineages, are sensitive to fungicides, and some are resistant to certain fungicides. So we've done baseline screening of fungicide resistance. And as part of a cluster I lead on emerging disease and global food security, I've been working with my collaborator, Qin Wei, and his student, Rajesh Paul, and another collaborator, Anna Whitfield, on developing some sensors for detection of P. infestans. And we're trying to develop sensors that can be used attached to smartphones using small microneedle patches to quickly extract DNA. And then you run the LAMP assay on the phone and you get an answer on whether it's P. infestans or not. We've also scaled this up to other tomato pathogens. And then we're developing volatile sensors that are based on volatiles emitted from P. infestans and infected plants. And we can separate healthy from diseased plants by using volatile profiling. I'm just going to show a quick picture of what the smartphone device looks like, but essentially this is beta test version 1 of using these four samples can go on the slide. They're lyophilized reagents, and essentially it's an in-field LAMP assay that will light positive or negative based on whether it's P. infestans or not. We've run this out for tomato spotted wilt virus as well, and we're working on a couple of other pathogens. For tomato, bacterial, virus, oomycete, and fungal pathogen. And ideally, we want to pull all this kind of information together into improving data analytics and disease surveillance for plant diseases. And this is not just for plant diseases. These same kinds of tools can be used for disease surveillance of human or animal diseases by studying trade networks, how people or plants move, by studying climate factors, using early disease detection, using DNA analysis, sensors, mining historic herbaria or data from herbaria, and disease identification apps. And the goal is to move these sorts of tools into a surveillance system that we're calling a plant aid database. We have a team funded here at NC State, a cluster of faculty and affiliated faculty that received a grant from the university's plant science initiative to improve disease surveillance of tomato pathogens, but it's scalable to other systems as well. And with that, I'm going to end and just thank my lab, who's shown here in front of our new plant science building, which we moved into a few weeks ago, and a number of different funders of the work. And I'd be happy to take any questions. And Sandy, I think you're going to.
Yep, that's my job. Thank you so much, Jean. That was absolutely amazing. What I love about your work, and I've always loved it about your work, is the way you can integrate, from the past into the present, and then actually kind of show us how we can use all of that to predict the future, which is really what we need to do if we're going to save the planet, isn't it? Is to use the past to predict the present and predict the future, which is just amazing. So we've got some questions. So our first question is, and I think I'm interpreting this correctly, is that does the nucleotides of the genome, and I'm wondering, so maybe the question is being asked from both the perspective of the parasite, but also of the host, of the plant, does this change with disease? Does the disease change the genome of the host, or does the genome of the pathogen change as disease progresses?
What we know from some of the published work on host-parasite interaction is that the genome of the pathogen is changing in response to the R genes that are being thrown at it through resistance breeding. So You know, and I would say that it's more the genome of the pathogen. There's an arms race between host and pathogen, but I would say it's more the pathogen genome changing. But you know, I could be
convinced otherwise.
exactly. Yeah, let me put it that way. That's a good answer.
We need to investigate that.
Exactly.
There's a question here about some different species of Phytophthora, but a couple of species of Phytophthora which were first detected in the UK with rhododendrons, Phytophthora amorum and Kernovii. And there was concern at the time that non-natives might cause a major conservation issue in native habitats. So Rhododendron ponticum is a non-native plant. Do you know if these or other Phytophthora have caused conservation issues as well as human food security issues?
So I'm not sure exactly what the questioner is meaning by conservation issues, but definitely remorum in wild areas. We're doing a lot of surveys in the US, native rhododendrons in the mountain areas in the east. There are issues of Phytophthora spreading from ornamentals into those areas and could destroy native biodiversity. So
I think that's what the question is aimed at, is you know,
yes.
And so it's clear that there are those kinds of issues as we move plants around, as you've so clearly shown through your research.
Yes.
Somebody's posted a question about Sarpo varieties, and Sarpo varieties are these Phytophthora late blight resistant varieties of potatoes, which we get here in the UK. They're made in Hungary. They're absolutely amazing. I've grown them in my garden.
Shaw's work. Yes,
yeah,
I know about
absolutely great. And so have you looked at these? And the answer is probably yes.
No, I haven't, but I know all about it from David Shaw's work. But we haven't tested Sarpo.
Okay.
But I know it's supposed to be really resistant. Yes.
I don't know. I never get late blight in my garden, and I grow Sarpos sometimes. So who knows? We'll see.
Yes.
But they are supposed to be resistant. Somebody else has said that they were surprised that they discovered recently that late blight was a problem in Scotland during the 19th century as well. So how much is known about the late blight problem in Scotland?
Oh yeah, well, immediately in September of 1845, Scottish growers in the Highlands were saying their potatoes are dead. It's been there a long time. There's a lot of the reports in the Gardener's Chronicle. If you read those reports back, it was in the Scottish Highland area where potatoes were grown as well. It spread throughout the whole UK and over to Ireland.
It's a monster, isn't it? And I mean, the thing is, Padma has put in the chat the link to the Gardener's Chronicle, which is on BHL, which is absolutely fascinating for thinking about plant introductions.
I get so sidetracked when I start looking at that. I just want to look at the ladies' clothing from the 19th century.
I know, it's brilliant, isn't it? It's so great. Someone else has asked us, what is the current thinking about the role of oospores? Are they more common than previously thought? And what is the role of oospores in all this diversity of infection and species and stuff?
So Manning Gallagly, who turned 99 this year, Manning Gallagly and his colleagues Galindo and John Niederhauser were involved in describing oospores in Mexico in the mid-20th century. And in many areas of the world, oospores were not found prior to that time. Anton de Berry in the 1870s was trying to understand the role of oospores in disease, and Clinton in the early 20s, G.P. Clinton, 1920s. But oospores, both mating types were found in the Central Highlands of Mexico, and they're produced there. They're now also play a role in Europe and the Netherlands. There's been for 20, 30 years reports of both mating types in the Netherlands and in Scandinavia. So oospores are playing a role in survival of the pathogen in these areas. And when the pathogen sexually reproduces, the next generation is going to be genetically more diverse than if it was clonally reproducing. So oospores are a survival structure that is causing problems in Europe and Scandinavia right now.
But it's probably great for the pathogen.
Exactly. And in South America, the mating types are, like in Peru and Bolivia, the mating types are spatially, geographically segmented. They're not in the same areas. You'll have both mating types, but because of certain mountain ranges and geography, it's not a sexual population. And also in South America, some of these areas, there's always a host available to cause disease. So it doesn't, I think about the pathogen just not needing to overwinter. in some of these areas.
Because there are wild potatoes growing all around.
Exactly, too.
Mhm.
That's true too. When I was down there with Tina, you know, we were like, there were wild potatoes all around those fields.
Oh yeah.
And the pathogen can overwinter.
Oh, that's really interesting. I mean, that's an interesting dynamic, isn't it? How that's going on between wild and cultivated and proximity, which we don't have in Europe, of course, because we don't have any wild potatoes in Europe.
Yeah. Yeah, just volunteer weeds and volunteer solanums that might be, or call piles, allotment gardens can be a source if someone doesn't cut down their tomatoes and overwinter them. They showed in the Netherlands that folks' allotment gardens could be a source of inoculum.
Interesting. Someone has asked what I think is quite a complicated question, Jean, so you can deflect this if you want, but it might be good to just have a quick kind of overview. Is they've said, what is the basis of plant resistance to Phytophthora? Can you breed strains that have this resistance? So you mentioned R genes, and this may be a question about how do R genes work?
Yeah, well, I will say at the outset, I am not a potato breeder, so I don't know everything there is to know about R genes, but I do know that R genes are being pyramided from different solanum sources to create a more resilient potato that will resist being overcome by the pathogen.
So I guess
breeding work being done, yeah, with different pyramiding of R genes from different host sources.
So just explain a bit about what R genes are. So R genes are the way that the potato resists the fungus, isn't that right?
Right.
Yeah.
The when the effectors of the pathogen are produced, they bind. If there's a resistance gene, it will bind with the the effector will bind with the R gene, and disease does not occur. There's a recognition between the pathogen protein that's secreted in the host and the resistance gene that's in the potato. There's like, think of it like an antibody antigen kind of reaction where they bind, although I'm just giving a general kind of explanation.
Well, it's quite complicated, but yeah, you're right. Yeah. It's like an antibody antigen kind of thing. So it's our genes are a complicated thing, but actually plants have mechanisms to overcome. So it is, as you said earlier, it's an arms race.
Right.
Yeah. Right. Somebody has said they've heard that a major contributing factor to the Irish famine was that they grew lumpers, which is shown to be exquisitely susceptible to blight. Is this so? And I mean, I think one of the things about potatoes, which I find really interesting, is that they're, of course, vegetatively reproduced. So when you vegetatively reproduce something, your whole field is genetically identical. So it doesn't matter what variety it is. It's the fact that it's genetically identical. Isn't that true?
Monoculture of the same type. Exactly. Mm-hmm. It's like they're clonally reproducing vegetatively. Yes. And lumper, well, everything that was planted at the time was susceptible. No, actually within maybe 3 or 4 years, breeders were working on something called Champion, which is supposed to be like the blight blasting variety, but it was once again overcome. But lumpers were very susceptible.
Yeah, and everything was lumpers because people saved their potatoes from the year before. So it wasn't because they were stupid in growing lumpers. It was because that was the survival mechanism, wasn't it?
Exactly. And interestingly, Darwin, you know, he had tenant farmers on his property, and he was paying them in potatoes, just like in the landholders in Ireland.
Oh, that's interesting.
His wife, Emma Wedgwood Darwin, she was part of the Wedgwood family. She actually bought meal bread tickets and gave the folks on the farm these tickets so they could buy food because they didn't have the potatoes. So she was trying to help
out with the problems.
Feed the workers on the farm.
Yeah, that's really amazing. Somebody's asked, is admixture, and I presume this means kind of admixture of different potato varieties in a field, linked to increased virulence, or no, well, admixture of strains of Phytophthora, I suppose. Is admixture linked to increased virulence or adaptive evolution in Phytophthora infestans?
So admixture, when I used the term admixture when I was showing some of our Illumina data on the P. andina and P. infestans, the genomes are There's admixture of two different genomes. So there must have been an ancestral host that they shared.
Mhm.
So there are Phytophthora species that hybridize with other Phytophthora species, and you can get more virulent types. So admixture, there are a number of studies going on now to look at that. And I think with P. infestans, I don't know that it's been clearly documented, but it's possible, I'm sure, because P. infestans will mate with other Phytophthora species.
Yeah, so it's a potential increaser of virulence, having lots of
Yeah.
different strains together.
They have to be opposite mating types to mate.
Mm-hmm.
Yeah, and a lot of times there's either one or another mating type circulating in the populations. But within P. infestans, you'd have to have both mating types to get.
So does that mean that a lot of these Phytophthora, because you were talking about sexual reproduction in oospores earlier, so a lot of these different Phytophthora kind of are clonally moving about?
Exactly. In tubers, they're moving about clonally. But in some areas, like I mentioned, in Scandinavia and in the Netherlands, they've got multiple mating types in the same field, and they're getting genetically diverse new populations at sexual reproduction. So I mean, you could call that admixture of genomes of P. infestans.
Yeah, possible. So someone has actually put a really interesting comment in here is in the original outbreak, John Lindley, who was editor of the Gardener's Chronicle, as you know, invited readers around the UK and Ireland to write in about the state of their potato crops to track the spread of the disease. Do you see this as a predecessor to your disease surveillance and citizen science activities?
Very interesting. Yes. Yes, I would say so. That's really, I have Lindley's history sitting on my shelf here, and yes, and he was not at all convinced that this disease was caused by a pathogen. He and MJ Barkley got into a big debate on that.
Ooh,
so
interesting.
Yeah, so it's
he was trying to disprove Barkley in a way by his kind of doing that. Oh, that's interesting.
Yeah,
but it is an interesting thought that that's a way of getting everybody involved in thinking about something that's happening in nature, isn't it?
Yeah, so they were writing in, I mean, there were weekly reports, and they were just a paragraph or two. Very eloquently written, and then the grower or the Lord would sign his name and where they were located. So yes, that was really early disease. We actually want to mine the Gardener's Chronicle stuff. We've only looked at the patent reports on the US side, but there's a whole wealth of information, and now a lot of them are in PDFs.
Yeah, no, it's because Gardener's Chronicle is on BHL, of course, the Biodiversity Heritage Library, of which the Linnean Society is a big contributor. So that's absolutely fantastic. That is just really, really interesting. Let me see if there's more questions that are coming, but actually I'm going to ask you a question, Jean, is what do you think will happen next?
Well,
terrible question, I know.
Next, in terms of, I know what I would like to do next in terms of our research.
But what's going to happen next with potatoes?
Oh, you mean you want on the host end? Or
yeah.
I'm thinking about on the pathogen end.
Well, both ends. We can go from both ends.
Yeah, I think there's going to be more studies done looking at changes in the host in response to the pathogen, like one of the questions earlier.
Yeah.
Looking at how our gene deployment might have driven evolution of the pathogen. Those kinds of questions using genome sequence data. We're actually, my student Allison Kumler is working on some of that right now. We're also more exploration. A lot of materials come out of Mexico. In fact, the Solanum demissum is in a lot of the modern grown potatoes that are out there. And Robin Buell published a very interesting paper on the evolution of potatoes, and like only 14 to 15% of all the potential genetic diversity that is in modern potatoes from ancestral wild types in the Andes. So we've just kind of at the tip of the iceberg and exploring what kind of host diversity might be deployed to control not only late blight but other diseases. And these whole genome sequence papers, all these solanums are starting to tell us that there was like a favorite variety, and then that was the predecessor of the next one, and they kept carrying the same our genes down for generations. So
Yeah.
I think the breeding is being informed now more by these whole kind of population genomic studies on the host.
Yeah, and as we understand more about potato phylogeny as well, about the phylogeny of those particular taxa, that will help as well, I suspect.
Exactly. And then just conserving, I know you've been very involved in conserving biodiversity. As climates change in the Andes, high temperatures are impacting what growers can grow and where they can grow, where they have water. We have to really be cognizant of conserving that biodiversity so it isn't lost before it's even discovered.
Well, absolutely, absolutely. Well, I think that's we don't have any more questions, which I mean, I could keep asking you questions all night, Jean, as you know, but thank you so much for coming along and actually for ending on a really upbeat note about one of the main values and missions of the society, which is about understanding and conserving the diversity of nature, because that's what the Linnean Society is all about. And I think you've really eloquently demonstrated how we can go from what seem like just ancient herbarium specimens preserved on pieces of paper to actually something that has a real-time use in helping solve real-time problems today. So thank you so much, Jean, and I look forward to seeing you when you come to Ireland and meeting again soon in person. Thank you so much for a wonderful lecture.
We'll have to have an espresso coffee.
Absolutely. Thank you so much.
Take it easy.
Good night, everybody. Thanks a lot for coming.
Bye.